else?
I love it.
Oh, Christine, we're good.
YouTube's good.
All right, YouTube. Hello, folks. Going to start recording in the room.
Okay.
Record to
that's
the
awesome.
cloud. All right, welcome everyone to this week's Wetlands and Watershed Seminar. We are in the room and online and on YouTube. Thanks everyone for being here. I'm going to hand over the introduction to Christine. Just before we do that, I'll get this going so we all know that it's working. And all right, camera's here, so you can go ahead and do the introduction,
Okay.
Christine.
All right, great. All right. Hi, everyone. Nice to see you all this morning. One quick announcement before I present the speaker, which is that Dr. Mark Burtonus, who studies some of the foundations of the work that Joe's dissertation is built upon, although he added a lot of flair and color to what Mark did. But anyways, Mark Burtonus will be here giving a seminar on why experimental ecology matters to conservation on Friday of this week from 3 to 4 p.m. And that will be offered, he'll be here in person. So if you want to be here in the Phelps lab, you can be. But there's also a Zoom link that you can tune in. And there'll be a happy hour that follows that at First Magnitude from about 4:30 to 6:30 p.m. on Friday evening. So Charlene, if you can drop the link into the chat or the invitation, that would be wonderful. So without any further ado, I'd like to introduce Dr. Joe Morton, who's joining us very recently here at the University of Florida Community in the Center for Coastal Solutions. He'll be working a lot on sort of dune restoration and generally applying some of the kind of ecological principles to informing how we may better sort of enhance the recovery and resilience of our coastal landscapes. Joe's joining us from Duke University, where he did his PhD under Brian Silliman, who used to be a faculty member here at the University of Florida, and Duke sort of stole him away a number of years ago. So Joe and I share a sort of an academic lineage, but I want to give him as much time as possible to share with us some insights about parasites and their role in salt marsh structure and function. So thanks a lot, Joe.
Thanks for the introduction, Christine. Let me get the pointers.
Oops, sorry.
Oh, okay.
Okay, go ahead.
Thank you so much. Well, sorry to give this talk right before lunch and ruin everybody's appetites by talking about parasitic plants. It is what it is. So I'm going to be talking about a lot of work that I've done over basically the past decade about parasites and their roles in salt marshes, little things that do big things. Before I take you to this nice vegetated wetland, can I use this? I'm going to take you to a less, another vegetated ecosystem, but considerably drier. So you're out on the African Serengeti. It's pre-1965. So the rinderpest virus, this pernicious virus that infects a wide variety of ungulates, is exerting pretty heavy top-down control of this ecosystem. Rinderpest kills in mass ungulates like wildebeest, and wildebeest die. They're going to die.
Put it that way.
How's that? Oh, I'll just that works.
Okay,
That works.
I think if you click now, you should be good.
Okay. I'll click with this.
Yeah.
We can't wait.
Oh no!
Yeah.
Trying to kill these. Whoa,
Come on, do it. Yeah.
Bronzos. All right. So when wildebeest die, they're not exerting as much grazer pressure on the system, so grasses grow, and more grass means more fuel for fires to burn. Fires rage across the Serengeti. During this period, before rinderpest is effectively eradicated in 1965, the Serengeti, something that we might think of as a carbon sink, is actually a carbon source. It is producing more carbon than it's taking in. Now, in 1965, like I said, rinderpest is eradicated. There's a huge population eruption of wildebeest. They control the grass, and then woody tree growth is facilitated. And the Serengeti once again becomes a net carbon sink. Now, I love to tell this little parable because I think it is illustrative of the tremendous power of very small things. The rinderpest virus is orders of magnitude smaller than the organism that it infects, and yet it can have effects as fundamental and powerful as the carbon flux in an ecosystem, growth of woody trees. Now, this used to be a much more, I think, dramatic story, but now that we've sort of lived through a pandemic and we understand that a disease can affect the structure and functioning of our own lives, I think when I say that parasites can modify the structure and functioning of ecosystems, it resonates a little bit more. And parasites can do a whole lot of things. They can have pretty profound effects on individual hosts. It's not fun to be a host, as we all know. They can influence population dynamics of organisms, and they can modify ecosystem structure and functioning in two primary ways, either directly by controlling host density, just like that rinderpest example, or indirectly by controlling a suite of host traits. And you are looking at just some really gross pictures. Again, sorry about before lunch. Some really gross pictures of some of the ways that parasites can modify the traits of their hosts. Leucocorium paradoxicum, this trematode, can get inside the eye stalks of snails and pulsate in synchrony, makes the eye stalks look like a little grub. Birds will swoop down and eat the eye stalks, and then the parasite's life cycle is complete. Rhizocephalan barnacles and crabs, they control the crab's behavior, putting out a root network through their musculature and nervous system, changing their hormones, doing all manner of things, and hijacking their reproductive system. These are some really dramatic examples, but for the most part, parasites, when they modify behavior, it's usually a bit more subtle, a change in a rate of some activity, something like that. And we might expect that organisms that are infected with some sort of parasite are likely to act a little bit differently than organisms that aren't. And if the organism in question is abundant, ecologically influential, an ecosystem engineer, a foundation species, something like that, then we might expect wide-ranging ecosystem level effects. So now I take you to the more familiar grassland that I've worked in, southeastern US salt marshes. So in southeastern US salt marshes, if you've ever been out to one, you might have seen an area like this. This is a Dieoff area. Now, Dieoff areas are caused by a variety of things. But if you go to some of them, you'll often see this along their borders, large aggregations of this snail, Literaria irradiata, the marsh periwinkle. And snails, these snails participate in a fun little proto-farming mutualism. They gradulate the grasses and can have pretty profound effects on plant growth. And so to give you an idea of how a lot of these die-offs form, there's oftentimes, there we go, there will be a drought. This will stress out plants, and then grazing snails will take advantage of these weakened plant defenses to cause cascading vegetation loss of the marsh, opening up the marsh and forming these consumer fronts that can then propagate outward, taking lots of healthy vegetation with them. Now, snails are a major food item for a lot of critters, crabs, blue crabs, mud crabs, some birds, terrapins. Oftentimes, though, in these die-off areas, these organisms are conspicuously absent. Very stressful environments. Oftentimes, during drought years, you get reduced abundance of predators for other reasons. It's a different story. But there's something else that's eating snails, and it's eating them from the inside. And that is what I'm interested in. So I'll introduce you to one of my favorite organisms. This is Pororchis acanthus. Pororchis acanthus is a digeneian trematode. And for those of you who are perhaps not familiar with the digenous nematodes, these are flatworms with elegant life cycles, elegant and complex life cycles. They spend their adult stage in a vertebrate. Vertebrates will release eggs into the environment. Those will then hatch into neuracidia, which infect snails. Once they're in that snail, they are parasitic castrators, so they will consume the gonad of the snail, replace it with parasite, asexually reproducing tissues, and they just kind of turn the snail into sort of a factory for cercaria that then will swim out of the snail to infect a second, sometimes a third, fourth, fifth, intermediate hosts. And these are called trophic hosts because they have to get eaten by the final host in order for the parasite to be transmitted. So complicated life cycles. Parorchis acanthus is a parasite that's actually fairly common in marsh periwinkles, but it's only common around these Dieoff areas. I'll talk a little bit more about that later. It's a fairly cosmopolitan parasite too, as parasites go. It infects a large number of birds. Pretty much every coastal bird that you crack open, you'll find Parorchis acanthus and the Cloaca and bursa facultative. That's an infected snail there. You can see the gonad that's populated by trematode larvae. So I got to thinking when I first found these infected snails prior to starting my master's, well, maybe they're doing something to the snail's behavior. These are ecologically influential organisms. Perhaps they can alter their ecologically influential behaviors. So I investigated that. So two things: snails munch on grass, producing these wounds, gradular wounds, and they also do circutidal climbing to escape from predators. And parasitism will affect both of those things. So they will reduce the amount of grazing that these snails do. And they also, a good way to find infected snails is to wait for the tide to rise a little bit, and then the infected ones will almost always be submerged. That's probably for those cercaria to swim out and get to the next host. So I was finding in that's me when I was young. So I was finding right around these Dieoff areas tons and tons of these infected snails, and so I got thinking, well, maybe these individual level effects on behavior could ramify upward to affect ecosystem resistance to this drought-associated overgrazing that happens. So I did a tedious manipulative field experiment where I collected thousands and thousands of snails, and you can determine if they're infected or not by putting them in a little dish of water. The cercaria will swim out. You can identify the species, mark them with a little dot to confirm that they're infected or not infected. And you do this a lot, and you don't lose your mind, and you eventually have enough material to do a manipulation with. I wanted to see, right, how parasitism at different prevalences might help protect that marsh. I also manipulated salinity. Soil salinity is kind of a drought proxy there. So as you can see, the more parasitism that you have, the more vegetation is retained, the more growth that you have. I'll just show you a picture of some representative plots. So, right, drought conditions where snails can graze a whole bunch. When there's low parasitism, or no parasites in the system, the snails basically after 3 months cause wholesale destruction of the marsh. But when parasitism was sufficiently high, 30% infection prevalence or so, you got to retain a lot of that marsh, and with it, all of its functions and services. And then subsequently, did some fun manipulations on actively expanding Dieoff borders where we manipulated parasite prevalence. And parasites effectively slowed the rate of ecosystem decline. So parasites are kind of playing the role of predators in these systems by effectively reducing per capita grazing rates and then slowing down the rate of ecosystem decline. So then, I mentioned that these things are pretty common in die-off areas. I did a survey of die-off areas along the East Coast and found that infection prevalence could be pretty high in a lot of these die-offs. It was basically non-existent outside of that, though. There we go, there's me again. So one of the things I was interested in, though, a lot of the places that I had surveyed, they tended to have pretty consistent densities of these snails. But grazer densities can really vary in space and time. So sometimes you go to a die-off and it looks like that. Sometimes you go to a die-off and it looks like that. Tons and tons of snails chomping down on the grass. So I asked, maybe there's some context dependency to the ability of parasites, to facilitate grass and increase ecosystem resistance. When grazer density is very high, the effects of parasites and the ameliorating effects that they have are likely to be swamped. When grazer density is very low, you may not see the effects of snails at all, and so parasite effects wouldn't be detectable. So I did another tedious field manipulation, this time with my colleague Sam Hub. We manipulated host density over a pretty wide range while keeping infection prevalence constant to see at what level of host density we would get be able to detect ameliorating effects of parasites. And in brief, this is the result of that. So uninfected snails, they tended to yield net losses in net primary production at around 67 snails per plot area. Infected snails, you get that net loss of biomass at a density of 84 snails per unit area. And then in between treatment comparisons, we found that parasites had this significant positive effect at these kind of middling densities. So just as we thought, at really high densities, right, the ameliorating effects of parasites are likely swamped. And then at low densities, they're just not detectable at all. So there's some context dependency there. Then I got interested in this problem from the supply side. So the parasites coming into the system, how does that influence the effects of parasites? So I mentioned, right, in these die-off areas, that's where you get very high prevalence of parasitism, but you don't get it in outside healthy marsh. And that is because these die-off areas are very, very attractive to the final hosts, birds. The presence of the final hosts is the big predictor of presence in intermediate hosts. So these die-off areas in the middle of marshes, they're very attractive foraging areas for birds. Birds will hang out there, munching on worms and fiddler crabs and things. It's a nice area for them to chill out in. So I asked, could these birds influence marsh ecosystem resistance to die-off by acting as the dispersive vectors for these parasites that control grazing in the system? So I did some photographic surveys at these 10 marsh sites with die-off areas along the North Carolina coast. And at each of these sites, I quantified bird usage using some camera traps. This is a very compressed video where you can see some birds foraging on a die-off at the rising tide. At each of these sites, I also quantified parasite prevalence and then the rate of die-off expansion. You can mark borders, the grass dies, you mark it again, it's very easy. These are all of the birds that we found and Dieoff areas. Tons of different representative species, bunch of white ibises and plovers out there. And then we also compared, we did some photographic surveys of adjacent healthy marsh, and birds are of course way more abundant in these Dieoff areas. Prevalence of parasitism increases with bird usage at a particular site, the bird density. And then the rate of ecosystem retreat declines with increasing infection prevalence. Some good correlative evidence that birds are helping increase marsh ecosystem resistance by dropping off these parasites. So then I did an experiment to confirm what I was finding from these surveys. So I built these big old cages out in the marsh, as marsh ecologists often do. And some of these cages I surrounded with a bigger cage to keep the birds out, so that these cages with uninfected snails inside would be relatively more exposed to the Parasitism swimming out of bird feces that's deposited in the Dieoff area, like so. So then, while I was letting that run, I did a bad experiment. Bad in that it was gross. So I wanted to test some alternative hypotheses, right? Because birds, they're also depositing a large amount of fecal material in these Dieoff areas, which has nutrients in it. Nutrients can increase plant growth. So I went out and collected, frankly, an obscene quantity of bird guano and sterilized it, right, to get rid of all of the parasite eggs, and then reconstituted it as a paste, and then dispersed it at ecologically relevant densities and plots along actively expanding Dieoff borders. That didn't do anything at all. I did that for months. That didn't change the rate of ecosystem retreat.
It changed
one
you.
iota. It did change. It was a rough time. I also I thought that maybe another way that birds, right, might increase or facilitate plant growth is by aerating sediments. Walking around, plunging their long bills into the sediments. I followed, groups of ibis around with redox probes, and I couldn't tell any difference. So back to the experiment. So here are some results. So exclusion worked, right? Trematode infection was increased in these exclusion plots dramatically, basically non-existent, or basically non-existent in exclusion plots. And then that increased prevalence corresponded to decreased snail grazing, measured by grazing scars that you can measure on individual blades of grass. And then the rate of ecosystem retreat in turn was faster in those plots where birds were excluded. So it looks as if, right, this interspatial exchange of these trait-altering parasites by birds can produce positive outcomes for ecosystems that are being altered by the synergistic effects of drought and grazing. It's cool stuff. And then my life changed. So this grainy picture is a marsh out on the Outer Banks of North Carolina. It's a place called Gunning Hammock. There's a lot of duck hunting that goes on around there. I went out there with a colleague who was doing some seagrass work in an adjacent area, and I thought I would collect some organisms for fun. When you're a disease ecologist, you always take yourself stuff home to crack open and open your Christmas presents later and see what you find. So I got some mussels and I got some snails, and I found some things inside of them that I hadn't seen before. Inside of the snails, I found Redia and Cercaria of a type that was unknown to me. This is a type of reniculid trematode, so it finds its way into the kidneys of seabirds. And then inside mussels, I found these metacercaria, right? This is what these turn into inside of the digestive gland. So I got to thinking that maybe these two things were probably the same organism. I did some digging in the literature and found one mention from a paper back in the 60s of this parasite that infects Literaria and has a weird life cycle. Like the Cercaria swim out of the snail, and then they insist back into the visceral mass of the snail. And then the snail is presumably eaten by something like a clapper rail, a snail-eating bird, and the circle of life is complete. And I did find those metacercaria inside of snails from this site, but I also found that if you exposed mussels to these cercaria, that they became infected. So it appears that this parasite actually preferentially insists in the digestive gland of mussels. And I did a bunch of field surveys, North Carolina, all down to Florida, and found that in some of these marshes, especially on barrier islands, prevalence was super high in snails around Dieoff areas. Additionally, the infection intensity in adjacent mussels was really high. You'll never eat mussels again, I'm sorry. So 5,000 parasites sometimes in a single mussel. I think the highest actually now that I've found is something like 8,000. That's what that looks like under a microscope. So each one of these little white dots that you're seeing is a little encysted worm. The unhappy bird that would eat this mussel is about to get a huge load of parasites. Nasty stuff. There's that up close. So I got to thinking about this because mussels are very important. This is a lot of the work that Christine has done. So mussels, they form these aggregations in the marsh, and they participate in a facultative mutualism with marsh grass. Mussels, having a mussel attached to you if your grass is kind of like having a little fertilizer factory. The grass shades mussels, it gives them a place to hold on to with their visceral threads, and then the mussels produce nutrient-rich biodeposits that increase plant growth. They're also biodiversity hotspots. So tons of crabs and other crabs and snails and things that you find in marshes. They like to congregate on these mussel mounds. There we go. Some figures from some of Christine's papers. Mussels are also important to marsh ecosystem resilience, to drought. So oftentimes when there is a drought that sweeps through the marsh, it will kill off a large number of plants, but areas that have mussels kind of pave over the marsh. They allow for increased retention of water, decreased soil drying, and plants can then survive and become kind of the nuclei from which the marsh can then regrow. So it's very cool, cool interactions. There's what I mean by the nuclei, right, for marsh regrowth. So I got to thinking, these parasites that are common in mussels and die-off areas, maybe they're changing mussel behavior that is ecologically influential. One of my students, Brian Davis, did some experiments, took mussels of an initially unknown infection status, and then subjected them to a biofiltration assay. We figured out that the more parasites inside of the mussel digestive gland, the less chlorophyll A they're filtering out of the water column, and the fewer biodeposits they're producing. That's a big deal, right? Because that is what underpins that marsh facultative mutualism between Spartina and mussels. So, with our conceptual diagram, I thought that perhaps these parasites, by negatively affecting biodeposit production, might be influencing all of these other things, benthic productivity and biodiversity and various ecosystem functions, in addition to this spartina growth. So I decided to do another incredibly tedious experiment. So I gathered thousands of mussels, thousands of snails, and I figured out the snails that were infected with Cercaria. Okay, good. And then I exposed these mussels from a population that was known to be uninfected, exposed them to different levels of the infected first intermediate host, such that in the end, after a couple of months of exposure, their digestive glands look like this, or perhaps more appropriately, they look like that. So high infection, medium infection, low infection. This is sort of what their representative guts look like. And that high infection intensity, that's sort of similar to what you would see out on a standard Dieoff area with a whole lot of infected snails. So I took these infected mussels and planted them out in the field in a manipulative field experiment. And then I subsequently monitored changes to spartina growth and all of those cool functions that I mentioned earlier. And this is what that looked like. So at high infection intensity, these mussels really did not influence spartina growth at all. Fine, there we go. I'll give you an idea there. So yeah, there's actually no difference between a highly infected plot of mussels and a plot without mussels. They're not doing anything for grass growth. And at, yeah, that's the crux of that. So other stuff. There we go. Parasites also influenced a bunch of other things. So the more parasites there were in mussels, there was less change in elevation. You know, these mussel mounds are usually fairly high because of the production of biodeposits. They can kind of accrete over time. Parasites prevented that. They also influenced the abundance of a wide variety of mound-associated organisms. So juvenile snails and mud crabs, fiddler crabs, purple marsh crabs, and benthic productivity. So parasites are indirectly influencing all of these biodiversity metrics by changing the nature of this mutualism between the grass. And also some biogeochemical stuff. So parasites influenced redox potential, probably by influencing the number of fiddler crabs that burrow into the soil and allow for oxygen to penetrate into the deeper sediments. And then also, parasitism influenced mussel mortality out there, at least in highly infected mounds. So in these highly infected mounds, you would often find the evidence of dead mussels out there. So the good thing about mussels is you can kind of do a little bit of forensics and figure out how they died. A mussel that's kind of opened up like this is likely to have died of natural causes, whereas a mussel that's been crunched like this is likely to have been crunched by a predator. Very fun science. So we looked at the number of mussels that had been crunched and the number of mussels that had died of natural causes across time in those treatments. So this is mortality over time. You can see there's some big trends. There's this kind of bimodal distribution in mussel mortality that takes place during the coldest time of the year and the warmest time of the year. So potentially parasites are influencing mussel response to temperature extremes. If you look at the mussels that are presumed to have been crunched, consumed, Most of that mortality takes place in the warmer months when predators are active. Things like raccoons and rails and crabs. This actually might be an adaptive aspect of this parasite's life cycle. Some parasites, in the strict sense, you don't want to kill your host, it's your home, but some of these parasites that are in their trophic host are sort of waiting around for their host to kick the bucket so that then they can be eaten by scavengers that are the final host. So that might be what's going on, we don't know yet. And then if you look at the mortality of mussels not consumed, again, you see this kind of bimodal distribution and death. And I have a group of students that's working right now exposing mussels to extreme temperatures and then cracking them open and counting their parasites. And their results are forthcoming, but it's looking like the more parasites you have, that might alter your ability to withstand these temperature extremes. We've seen this in a couple of other systems. Parasites can actually influence the production of heat shock protein, things like that. Then I had some other students look at some other potential mechanisms to explain this increased mortality. So they looked at byssal thread attachment strength, right? Maybe mussels that are highly infected just can't hold on to the grass or to other mussels, and so they're more easily picked off by predators. And so they did some very fun experiments where they put mussels of an initially unknown infection status next to a little dowel, and then tediously counted their number of byssal attachments after a certain time period, and also the force that it took to rip those mussels off their attachment. And yeah, the more infected you are, the weaker your attachment strength is. Also, the more infected you are, the weaker your shell. So my student Taylor Walker invented this ingenious device, crusher. It's an incremental crusher. You put a mussel in there and then fill this with water, sometimes some weights. And then when it crushed, right, we would then dissect and determine the number of metacercaria in the digestive gland. And sure enough, more infected mussels have weaker shells, so that might leave them more susceptible to predation. So then at the end of all of this, I did a survey of a couple of Dieoff areas in North Carolina. And I surveyed infection intensity in mussels with distance from the Dieoff edge. And what I found was, first of all, at every site that I looked at, I found these things in mussels. And there's this decrease further away you get from the source, right, of those infections, these infected snails. But even in areas of the marsh where there are no infected snails, 40 meters away from the Dieoff, you still get a fair number of infections in mussels. And so there's actually this kind of infection halo that extends beyond the area, the disturbed area, potentially affecting the mutualism between Spartina and mussels, well outside of area where infection is most intense, which has some interesting implications for marsh ecosystem resilience. Oh, there we go. Okay, I just said. Okay, so I've come to the end now. So if you take nothing else away from my talk, I hope that I have impressed upon you that parasites, although small, can be very important, and that also they are worthy of us being the focus of manipulative experiments. Really, the last 20 or so years, this is sort of the golden age of parasite ecology. We're just now starting to incorporate parasites into food webs and explore what they're doing in ecosystems experimentally. The thing that has moved ecology forward. I'm giving a preview for Berkeley. So the experiments, the things that have moved ecology forward. And so manipulative experiments can be very difficult with these organisms, but where possible, they can reveal the long reach of very small things. And we might not be able to fully understand how ecosystems work without incorporating these organisms, which constitute the vast majority of life on the planet. Most organisms are parasites, whether you like it or not. They're everywhere. Okay, I'll shut up, stop talking. And if you have questions, please feel free to ask.
All right, so we'll start in the room and then we'll go to online. I know Sherilyn's monitoring the YouTube. So questions from the room to start.
Yes.
All right, I have a question about what makes, so you have these different sites that had different numbers of bird usage.
Yes.
And I'm curious, what makes a Dieoff site particularly attractive or less attractive to birds? Did you get any insights about
So
that?
the size of the die-off is a big one. I mean, that's just, the larger the die-off, the more birds it can accommodate. And then there's a big tidal influence. So if you look at when birds use these areas, 90%, I think, of all the birds that I captured from these photographic surveys were using the marsh surface in these die-off areas during the brief window where the tide is flooding and where it's draining out. Some of the longer legged birds, herons and things, stick around for a while and forage for mummy shells or something. But when the tide is flooding or when it's draining out, that is when it is easiest to see tilitrid amphipods and polychaetes and crabs moving around, and they're accessible. So I think that they use the marsh during that time. It's also the time when snails are probably the most vulnerable to infection because there is an aqueous substrate through which parasites can move, and they're more likely to be submerged in the first couple of minutes of the rising tide before they vamoose and go up the stalks of the grass. Yeah.
So you said that, like, this unlucky bird who's about to have this meal, but and maybe I missed it, but what are some of the physiological or what are the effects on birds that don't seem to overcome this sort of the benefit of feeding in these areas is food.
Yeah.
And then, but is there some maladaptive outcome from feeding on all this parasite?
I should say that birds have iron stomachs, so they're going to, not all of those parasites are going to survive a bird's stomach. That being said, birds can contain, seabirds can contain an absurd number of parasites, but they're quite small, and the energetic cost of having, I don't know, 1,000 little reniculate trematodes in you is probably fairly minimal in the grand scheme of things. There might be a little bit of a fitness cost, but yeah, it's not going to kill you, I suppose. I mean, these organisms, right? They co-evolved with their parasites, so they're good at dealing with being infected. Unlike ourselves, we started cooking our food, a couple thousand years ago, and that was a good choice.
Is there any behavioral changes in birds from parasitism?
Behavioral changes, there probably are some, depending on like the site of encystment. But yeah, I don't know what these particular parasites that I study are doing to birds, but probably not too much. They're mostly encysting in areas that are fairly inconsequential. The cloaca, areas around the kidneys, maybe the lumen of the intestine, something like that.
All right, go ahead, Mark.
All right, thanks, Joe. That was amazing. I am fascinated by this microscopic scale of influence on literally these ecosystems. And I remember taking a parasitology course one time long ago and being fascinated, but it's not really been in my forte lately. But either way, wonderful talk. So now the question, what's the temporal influence of these parasites? Like, I didn't get a feeling for how long some of your experiments were to show these influences. So just ballpark, are we talking days, weeks, months? Yeah.
So to give you an idea, if you're a snail exposed to a trematode miracidium, there's about 30 days before that parasite will effectively colonize your gonad and you start to display behavioral changes. Once infected, though, the snail is infected for the rest of its life. 0% recovery. As for the temporal dimension of experiments, most of these experiments are the length of the growing season, with the exception of the mussel experiment. That one lasted an entire year. Does that answer your question?
Yeah, close enough. I was just trying to get an understanding of what that kind of loop is, or the potential life cycle would be for those parasites. And yeah, you got it. Yep. Thank you.
Amanda, do you want to go ahead and ask your question?
Yeah, sure. Great talk. Thanks, Joe. The main thing is I was just curious of how you think these parasites might be affecting marsh resiliency and systems that say are not so much being affected by drought, but things like sea level rise, for example.
So in marshes that are not being affected by drought, if there's still die-off with snails, then yeah, there will be parasites that infect snails. They can potentially slow down the rate of die-off. For sea level rise, maybe not so much. I don't know if parasites can help out there, but alas.
Well, to the biodeposit production.
Potentially, yes. With mussels, yes. I'm thinking about the snails. Yeah. Yeah, potentially some effects on mussels. That answer your question?
Yes, thank you.
YouTube friends, we got a few more minutes left. So if you got a question, please drop it in the chat. We'll pass it along. Anyone else in the room?
I'm like curious about this halo effect on
Yes.
the mussels, and if there is really no recovery once you're infected,
Mhm.
that would suggest that at like a large spatial and potentially like a decadal time scale,
Mhm.
you would have lingering effects on sort of the dampened mutualism, right? Like,
Potentially, yes.
so it just makes me wonder whether it's worth going back to some areas that we know got nailed in the past
Yes.
and evaluating biodeposit production and the strength of the mutualism and whether that can explain
So
spatial variability and mutualism strength.
I can say anecdotally, I have been back to some recovered sites, and the intensity of parasitism is still higher than marsh that was unaffected.
Yeah.
So potentially there are these legacy effects of die-off regarding disease.
So you did an experiment with bird exclusion, but sometimes when you do marsh restoration, you also build bird exclusion just to get your plants growing, right? So like in the Northeast, we do goose exclusion fencing. And so, I mean, these are natural, there's a host parasite relationship. However, like, would it maybe be a good idea no, it'd be the opposite, right? Because, so if you were to replant an area of die-off and you excluded birds, now help me with the logic there, would that improve or decrease
Decrease expansion of, it would be a bad thing.
It'd be a bad thing, so never mind on that one. Okay.
So I'll tell you another anecdote. So some of the areas of the marsh outside of die-off areas where you do get elevated infection prevalence in snails are roosts. So areas where birds sit up in a dead tree or something and are raining down parasites onto snails beneath. Those areas tend to be rather healthy, not a whole lot of grazing going on there.
We got one more from Mark. Go ahead, Mark.
Yeah, so I'm curious with the prevalence of parasites, is there a parasite on the parasite? Like, is there a mechanism that controls the parasite at some point? If it's so prolific and does so well, what's its feedback other than potentially killing the host and losing its advantage there?
Right, so controls on parasites. So there's, other than the abiotic controls, I'm trying to think, in some of I didn't present it here, when I did that bird work, I did some additional modeling work where I included a lot of environmental parameters. One of the things that really shakes out is temperature. So these mobile forms of the parasite are pretty sensitive to changes in environmental conditions, and temperature is perhaps the biggest one. So potentially, right, if you get a lot of warming, that might negatively impact the transfer of parasites, at least from snail to mussel, something like that.
And is that temperature effect just a shortened time within which it has to find a new host, or is the temperature actually a direct mortality mechanism?
It's a direct mortality mechanism. Yeah, above a certain temperature, those mobile forms just die.
Got it.
Cool.
All right, well, let's give Dr. Morton another round of applause. Thank you very much. And yes, see the chat for the link for the 3 o'clock seminar on Friday. And otherwise, we will see you all next week. Thanks, folks.
Thanks, everybody.
Thank you.
Good job, Joe.
Thank you.
So such a pretty talk and